Hello everyone, welcome to Molecura. Today we are going to be talking about galactose metabolism, and we are going to go through every step and we are going to see what diseases are associated with galactose metabolism. The first step of galactose metabolism is going to be to get the galactose out of the lactose that we are eating. So lactose is going to come from dairy products like milk. So the first thing that we have is lactose. Now here we have shown it as 6 carbons, but it's technically more than that because lactose is a disaccharide. Lactose is going to be converted into two things, into glucose and into galactose. And this is the first step that is catalyzed by an enzyme that we call lactase. So lactase is going to be our enzyme here, and what we end up having is going to be one glucose that's going to go to this side, and of course after that we will have our galactose. And now our galactose is available to be metabolized by the body. Now the first step is going to be almost the same as we had before in glycolysis and in fructose metabolism. So this is going to be a phosphorylation reaction, which a phosphate group is going to be added to the carbon number 1. And the enzyme, same as the previous pathways, is going to be called a kinase. This time it's galactokinase. This enzyme, as the name suggests, is going to be using ATP, and this ATP is going to get hydrolyzed to ADP, and what happens is a phosphate group is going to be attached to carbon number 1, and what we end up having is going to be galactose 1-phosphate. It's a galactose molecule that has one phosphate group on carbon number 1. Now the importance of this step is to keep the galactose inside the cell. Again, the same thing that happened with glycolysis and with fructose metabolism. We want to keep this galactose inside the cell, and the addition of this phosphate group, which is negatively charged, is going to keep this galactose inside the cell. After this galactose can be converted into glucose, and how does this happen is going to be our next step. So here we have a molecule that's called UDP glucose, and UDP glucose stands for uridine, which is our nitrogenous base, diphosphate glucose. So we are going to have a pyrimidine here, which is our uridine. We're going to have our ribose sugar, and we are going to have two groups of phosphate group, and that's why we call it DP. So what we have here is that to our phosphate group there will be attachment of a glucose. So there is a glucose that is attached to this phosphate group. Now this is going to act as a glucose donor, so it will end up giving this glucose that is attached to it and bring it here, and take this galactose and bring it here. So how does this happen? This will happen in one step, okay, through action of an enzyme that is called galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase. And how does that happen is that we're going to have our galactose 1-phosphate here, and this is going to be converted into glucose 1-phosphate. So here we have our glucose 1-phosphate. And this is going to happen through this reaction here. And the enzyme that is going to be doing this is called galactose 1-phosphate uridyl transferase, or we can call it GALT, galactose 1-phosphate uridyl transferase. And what we'll end up having here is that this glucose here has replaced the galactose here, and it will come to here. At the meantime, the galactose that we have here, which is no longer in this equation, is going to be transferred into our UDP. So what we're going to have is a galactose attached to our phosphate, and the galactose will be here. So our galactose will be here. Furthermore, this galactose is going to be converted back into UDP glucose through action of an enzyme that we call UDP glucose epimerase. So UDP glucose epimerase, let's write it here. So here we have UDP glucose epimerase is going to convert back our UDP galactose into UDP glucose. And to make it complete, let's write the name here. This is our UDP galactose. Now, this glucose 1-phosphate that we have made here will obviously go to different pathways of metabolism into the cell. It can be shunted to glycogenesis, it can go to glycolysis, and it can go to, for example, pentose phosphate pathway. And for that to happen, it will need to be converted into glucose 6-phosphate. And that happens through action of an enzyme, which is called... So what we are doing here, basically, we are moving the phosphate group. Let's try to draw the phosphate group here. We had a phosphate group here. So what's happening here is that we are moving this phosphate group from carbon number 1 and we are putting it on carbon number 6. If you remember from the video that we had on glycolysis, the enzymes that are moving active groups within the same molecule are usually called mutase. So this is the same thing that is happening here. We have an enzyme that we are going to be calling phosphoglucomutase. So it's moving a phosphate group. The name suggests it's moving a phosphate group because it's a mutase. Where is it moving it? It's moving it on a glucose. So the name is going to give the function up. So what we end up having is going to be our famous glucose 6-phosphate, which we have already extensively talked about in the lecture on glycolysis. So this glucose 6-phosphate can be isomerized into fructose 6-phosphate, then into fructose 1,6-bisphosphate, and glycolysis can go on. So the metabolism of galactose is quite important because there are certain pathologies that is associated with it. And galactose metabolism is also important because that's the primary source of food when children are born. So galactose is going to be an important source of energy for the body. So this pathway will be happening in each and every cell to make glucose and ultimately make energy. Now, what are the pathologies that we can expect from this pathway? The first pathology that we can think about is going to be deficiency of the enzyme that we call lactase. If we have deficiency of this enzyme, we are going to end up with lactose intolerance. So here we are going to have our lactose intolerance. Lactose intolerance is going to have many different symptoms, and those are going to be symptoms like flatulence, bloating, abdominal pain, diarrhea, and in severe cases vomiting. So what happens is that lactose is not getting broken down into galactose and glucose. So what ends up happening is that this lactose is going to be consumed by the gut flora, by the bacteria that are available in the gut. And this is going to end up giving you a lot of byproducts of metabolism of those bacteria. So these are the bacteria in your gut that are going to be metabolizing the lactose that you are eating or drinking. So this lactose, this is going to be our bacteria. They are going to metabolize this lactose and they are going to produce a lot of gases, which includes hydrogen, which includes methane, which includes carbon dioxide. So all of these gases getting released here is going to end up giving you symptoms of bloating, of flatulence, abdominal pain, and diarrhea. So the symptoms are going to be of bloating, are going to be of diarrhea, and abdominal pain. So these are going to be symptoms of lactose intolerance. Now, the second disease that we're going to be talking about is going to be deficiency of galactokinase. So here we have our galactokinase, and we said that its action is to convert galactose into galactose 1-phosphate. We said that the importance of this step is to trap this galactose inside the cell. So if this step is deficient, what is happening is that this galactose enters the cell but is able to exit it without any problem. Technically, it's not going to cause many problems, and this is because our galactose, it's not getting accumulated inside the cell, so there will be no cell injury. So it's relatively a benign condition that we call non-classical galactosemia. So this is non-classical galactosemia. So this will be our second disease. The main thing that you're going to remember for this condition is going to be development of cataracts. So there will be cataracts, the hallmark of this condition. And the reason for cataract is accumulation of osmotically active substance in the lens. So what the patient will end up having is going to be cataract. We have spoken about this in the previous video of fructose metabolism. So what happens here is going to be a very similar situation. So what happens is that our galactose is going to be converted into galactitol, and galactitol is going to be an osmotically active substance, and as we talked about it, this is an alcohol. The reaction that's going to happen is going to be our galactose getting converted into a substance which is osmotically active, and this is called galactitol. And this all at the end of it is going to be telling you that we are dealing with a substance that is alcohol. As you remember it from the fructose video, is that this is going to be a molecule which has a hydroxyl group on every of its carbon backbone. The link to that video is going to be posted so you can watch it for more details. This reaction happens through the action of an enzyme that's called aldose reductase. So aldose reductase is going to catalyze this reaction, and there is going to be a use of NADPH in this reaction as well, and will end up giving us NADP+. Now, this reaction will end up causing accumulation of galactitol inside the lens and will end up causing cataracts, because this is going to be osmotically active and it will draw water excessively inside the lens, and the lens is going to get opacified and is going to get us cataract. So this is our non-classical galactosemia. So the question that arises is that what is the classical form? The classical form of galactosemia is going to be our GALT deficiency, which is our galactose 1-phosphate uridyl transferase deficiency. This disease is going to be causing a lot more problems than non-classical galactosemia. And the reason for this is, let's try to write the name of the enzyme here as well. So this is our GALT, and the deficiency of this enzyme is going to end up giving you classical galactosemia. So this is our classical galactosemia. And this condition is going to be very severe. Therefore, we have a screening in many countries for when neonates are born. They are going to be screened for classical galactosemia, because the main source of their nutrition is going to be breast milk, and which is going to have galactose. So deficiency of this enzyme is going to rapidly cause liver dysfunction. And the reason for that is that we have the lactose which the baby is consuming. Lactose is getting converted into galactose. This step is also intact. The next step is our galactose getting trapped inside the cell by galactokinase and producing galactose 1-phosphate. So this step is going to be defective, and glucose is not made through the action of GALT, which is our galactose 1-phosphate uridyl transferase. Now, when this does not happen, the galactose 1 phosphate has nowhere to go because it has already been trapped inside the cell. So the accumulation of galactose 1 phosphate is going to be causing the majority of the problems with classical galactosemia. So the symptoms are going to be of early jaundice. Technically, this galactose can accumulate in various tissues, including the liver, including the brain, the kidneys, the eyes, especially the lens. So the symptoms are going to be according to where this galactose is getting accumulated. So the first one is going to be of jaundice because of accumulation of galactose in the liver and ultimately causing liver damage. So this is going to be an important aspect of the disease. The second thing is going to be accumulation in the brain, will cause failure to thrive, and it's going to cause developmental delay. So we're going to have developmental delay. We're going to have kidney problems. The kidney problem is going to be very similar of what we discussed in fructose metabolism, and that is going to be cause renal tubular cell damage because of accumulation of galactose 1 phosphate. It's going to first deplete the phosphate inside the cell, thereby it's going to damage the active transport, and secondly, accumulation of galactitol is going to cause more problems such as osmotic injuries, so renal tubular cell damage. Of course, in the lens, it's going to end up giving us cataract. So cataract when it goes into the lens. Now, the child is going to present with nausea, vomiting, diarrhea. There's going to be failure to thrive, and these are going to be the initial symptoms that the children present with. And as it progresses, we're going to have jaundice, we're going to have hepatomegaly, and we're going to have liver damage. Another point to consider, which is going to be important, especially for exams, they like to ask about classical galactosemia and increased risk of E.coli sepsis in neonates. So neonates who are suffering from classical galactosemia are at increased risk of E.coli sepsis, which is going to be an important topic to be aware of. Increased risk of E.coli sepsis, especially in neonates, is going to be one of the features of classical galactosemia, which is the deficiency of galactose 1 phosphate uridyl transferase. So the treatment is going to be dietary restriction of lactose. So by this, we have discussed the metabolism of the three major carbohydrates in our body, namely the glucose, fructose, and galactose. In the next video, we're going to be talking about the TCA cycle and how it gets connected ultimately to the electron transport chain and ATP generation in our cells. Thank you for watching.